Okay. Okay. Hello, everyone. I'm Jianyu Yang from Mahoney Lab at Penn State. So I'm going to present how to apply a joint sequence and chromatin neural networks to characterize the differential abilities of FOC transcription factors to engage inaccessible chromatin. Sorry, it's a very long title. So as we know, the transcription factor is an important player in the gene regulation. So in recent years, there have been widespread application of deep neural networks to understand the transcription factor binding code. And there are many successful examples, including the previous talk. For example, the BPNet is a neural network that is able to take 1,000 base pair long DNA sequence to predict a base pair resolution or transcription factor binding profile. And so far, the largest model is very powerful called Informer. It can take 200 kilobase long DNA sequence to predict over 7,000 genomic tracks. This is a very impressive work. And all these models, most of these practices, they like to solely take the DNA sequence as input to predict the transcription factor binding. Although this makes sense because the transcription factor binding definitely wants the DNA motif there, and the convolution, for example, the convolution layer, this design is very suitable to scan all kinds of the DNA sequence motif and to give you the information of all the motifs of the transcription factor you are looking on or the other cofactors. But it misses the one important information about the transcription factor binding, which is the pre-existing chromatin information. So except a few of transcription factors, they are able to turn the pioneer factors, they are able to bind to their target sites without the help of without any prior context. Many transcription factors binding actually, they depend on either the pre-existing open chromatin or the histone modifications or the help of other cofactors. So this is an important missing pieces of application of deep learning neural networks to understand transcription factor binding. So we decided to look at this aspect. So what we try to do is look at this specific family of transcription factors called FOX family factors. This FOX family factors has a very well-known pioneer factor, FOXA1, and many of the members of this FOX family factors, they share a very similar DNA binding domain, and their cognate DNA motifs are very similar to each other. However, during the development, many of these factors actually, they play very distinct roles. So this suggests they might regulate different genes. This suggests they might bind to different binding sites. So we specifically look at these 5 FOX family factors, A1, C1, G1, L2, and P3. And we induce them in mouse membrane stem cell. And after 24 hours, we use ChIP-EXO to collect their binding sites. So in this case, this is a very interesting case that these all 5 factors, they share exactly the same pre-existing chromatin information. And we have the chance to take advantage of the rich data set existing in mouse membrane stem cells on chromatin information. And after this 24 hours, the ChIP-EXO data shows us among the top picks, actually these 5 FOX family factors, they have very distinct binding choices. So this suggests even under the same chromatin environment with a very similar DNA binding domain, they still choose to bind to different sites based on their preference. So the first thing we try to do is using a sequence-only neural network to see to what extent the sequence is able to explain this diverse binding site choices. So the model we use takes a 500 base pair long DNA sequence, and the design is very simple. It's like convolution tower with fully connected layer to predict the binding strengths of the transcription factors in the 5 base pair regions. So the performance of 5 individual models on these factors, they vary a lot. Some perform very well, like perform okay well, like FOXG1. The correlation is 0.79, and the worst one is the FOXC1. We first want to look at the sites I just showed to see how exactly how well our model, the sequence-only model, is able to recapitulate the diverse binding site choices. And on the left side is the true ChIP-EXO signal, and on the right-hand side is the prediction from the sequence-only neural network. And we can see that for FOXA1, it kind of knows that there are 2 or 3 clusters with the enriched FOXA1 signal, and some regions should have very weak FOXA1 signal. But for the remaining factors, especially like the FOXG1, it couldn't distinguish which side should have a stronger binding of the FOXG1, which side should have weak binding of FOXG1. So we further use a local feature attribution method. It basically tells you what exactly the model thinks is important in the input DNA sequence to look at what it is searching for and how does it able to explain the binding site choices. And we can see that the importance score is also consistent with the prediction. Only for the FOXA1, it's able to see that, okay, there's a very important features existing in these two clusters, and I should predict a strong binding of FOXA1. But for the other factors like FOXG1, it basically uniformly say all these sites are very suitable for FOXG1 binding, but actually it's not based on the two facts. We further run a global feature attribution method based on these importance scores, basically try to summarize the pattern the model is looking for. And these motifs tells us, except the TGTT-AC motif, I just showed a very standard cognitive motif for Fox family factors. The most frequent the motif is actually a TGTT, basically a repeats pattern here. So there's no indication of any cofactor that can explain why they choose the different binding site choices. So this is on the DNA side what the model can do. So how about the pre-existing chromatin? We first select histone modifications and ataxic signal to run a cron HMM on these all the histone the chromatin information and to get the chromatin state information and overlap these chromatin states with all 5 Fox family factors to get an overview of how is the association between the chromatin information and the Fox family factors. And we can tell even from the overlap percentage, there's a huge difference. Like FOXG1, FOXP3, they overlap with the active chromatin states a lot. And surprisingly, FOXA1 is kind of like in the mediate between among these 5 Fox family factors, although it's a pioneer factor. And FOXL2 and FOXC1, they are more associated with quiescent chromatin state. So it suggests that maybe there's some different association there. So the next step, what we want to do is to put this pre-existing chromatin information into the model we just mentioned to explain that, basically stat the DNA sequence and chromatin information and do exactly the same task to see how well it can predict the transcription factor binding strength. This is the chromatin information we used for the prediction. So after training, we have 2 types of models. The first row is the sequence-only models. The second row is the model with both the DNA sequence and pre-existing chromatin. And we can see that generally for every Fox family factors, adding the pre-existing chromatin information is very helpful to improve the model prediction. So which means even for the pioneer factors like FOXA1, it still needs some chromatin information like pre-existing chromatin contacts to bind to some of their binding sites. By comparing the predictions at the top binding site, we can also see that by incorporating the pre-existing chromatin information here, it greatly improves the prediction over the 4 Fox family factors that didn't do well when using a sequence-only network. And we can see there's a difference between the binding strength for FOXG1, FOXP3. This is very obvious. So we already mentioned the DNA sequence can only give a very uniform importance score across all the sites for FOXG1. So what exactly does the pre-existing chromatin do in this neural networks to help the prediction? We further look at the feature attribution score on all the chromatin tracks. For ataxic, it's the most obvious one. We can see that actually, like for this L2 cluster, the FOXG1 doesn't have a binding signal here, and the DNA sequence attribution is very high in this cluster. But because this region is devoid of ataxic signal, so the model learns that because it thinks the FOXG1 relies on a pre-existing open chromatin there to bind to these sites, and it recognizes it doesn't have ataxic signal, which means it should penalize these regions and cause this region actually finding the prediction by the sequence plus chromatin model to lead to it doesn't have any prediction in these regions. Similar behavior, we can also observe that on the H3K4 monomethylation and several other active chromatin marks. So this tells us like the pre-existing chromatin definitely is very helpful to predict the transcription factor bindings. Finally, I want to just show one simple case to see how the model takes advantage of both the DNA sequence and chromatin information to predict the Fox family factor binding. So in this region, So in this region, the FOXL2 bind to this region. We can see there's a strong chip axial signal here. And the model feature attribution on the DNA sequence, it gives a very high score in this region. Basically, it recognizes the TTTG, the repeats we just mentioned. And on the chromatin side, although it doesn't have any active chromatin marks in this region, the model knows that it's a small gives a small penalty in this region, but it's not strong enough to offset the important score from the DNA sequence side. So it still makes a positive prediction. There should be a strong binding of the FOXL2 in this region. For the same region, for the FOXA1, it doesn't recognize any important sequence attribution in this region. And also the chromatin side, there's no positive contribution. So the model knows that there's no FOXA1 binding here, which is consistent with the chip axial signal in the same region. And finally, for FOXG1, it doesn't have binding in this side. However, the sequence network is able to recognize, okay, there are many sequences I like, I should give it positive importance score. But finally, because it recognizes the FOXG1, it heavily relies on the open chromatin. It heavily penalized because there's no ataxic signal in this region, so which leads to a negative prediction in this region. So this is all my result, and I hope I already show you the essence of our project. So basically, the summary is the paralog transcription factors, they are able to diversify their binding targets with their differential abilities to engage in inaccessible chromatin. And we have shown that the neural networks allow us to understand the sequence and chromatin determinants of transcription factor DNA binding. And of course, the pre-existing chromatin is very important for us to understand the transcription factor DNA binding mechanism. So finally, I want to thank all other authors and colleagues in the lab, Sean's advisorship and other collaborators' work on our experiment. This is our lab photo, slightly photoshopped, and this is our manuscript. Please check out the full results if you are interested. Thank you for your attention.
We actually have lots of time for questions, 3 minutes. And I want to ask the first one.
Yep.
So the chromatin state is also, of course, itself a function of DNA sequence.
Yes.
Right?
Yes.
So how does your approach contrast with, like I use all the same data, but I use it as output and maybe use a slightly wider window?
Yes, I think you're right. Because I already mentioned actually those sequences only model. They use a wider DNA sequence window. They actually can do very well. But I think the problem is we can interpret how exactly that chromatin influences the transcription factor, right? Because on the performance side, we can see it's very well. But you try to explain that because the association between the DNA and the chromatin is like, I think it's by evolution, like slowly accumulated. You can't exactly tell like by importance score, like what exactly how the DNA sequence influence the chromatin, then influence the transcription factor. I think our method is better in a way, give a straightforward explanation, like how exactly, if in a cell, I want to induce the transcription factor, how exactly this chromatin state there will influence the transcription factor binding. I think this is more on the interpretation side.
The other question I had is, can you tell from the protein sequence whether a transcription factor will bind in heterochromatin? Is this like, what is this a function of?
Yes, that's quite interesting. We haven't looked into that, but these 5 Fox family factors, they don't look very similar to each other outside the DNA binding domain. Yeah, it is very interesting direction. If I can.
Is it a function of size?
Yeah, I'm not quite sure. Maybe there are some.
Okay.
We don't know. But it would be interesting if we use that as a accessory input for the deep learning model.
Yeah. Oh, okay.
So you did 500 base pairs in economic techniques, right? So if we shorten that to say 50 base pairs, do you think it will still work? Because mostly PFS sites are considered between 50 to 500 base pairs. So do you think this approach will still hold with the chromatin state information?
Okay. So the question is if we can shorten the DNA, the input window to 50 base pairs. I think that would then hurt the performance a lot because I feel like 50 base pair is not enough to 250. Okay. 250, I think it should still give a kind of okay performance because I think I have tried a model like that. But the point is, it should be big enough to cover both the transcription factors and the cofactors. like 250 is kind of like hits the limit of incorporating two factors in the same window. So I would always suggest the bigger the better if your GPU is able to hold that big input.
I guess that's kind of two different questions. So one of the questions I had with the Pioneer Fox family members versus the non-Pioneer members, is there any evidence that in terms of the protein domain, since you said they are quite different in like actual amino acid composition, is there any evidence that some of them can recruit DNA modelers to allow for changes in accessibility? And kind of go along with that, do you think that if you had assayed for the chromatin accessibility and those other markers at maybe 48 hours of just compared 24 to 48, would those Pioneer transcription factors have just altered the epigenetic landscape of those binding pockets where they're actually found at 24 hours?
First, I think like FOXP3, like these factors have been reported to interact with other factors. I'm not sure if they are DNA modifiers, but definitely you are correct. They work with other cofactors. And in terms of the 48 hours compared to 24 hours, we selected 24 hours actually is because in the experiment we found out the 24 hours is already the biggest change to the chromatin, the most change to the local chromatin state already happened. So I'm not sure the 48 hours we will be able to see more cofactors. Like, does that answer your question?
Yeah. So then these Pioneer factors, maybe not just Fox, but more Pioneer transcription factors in general, are binding to heterochromatin that remains in a heterochromatin state. It's not like it's changing the accessibility after it's binding or something like that.
Yes.
No.
No?
It opens. No, we did the test.
Yeah, it does open. I mean, sorry, I might misunderstood your question, but my point is these Pioneer factors does bind to their targets and change the local chromatin. And 24 hours already, there are many regions have been opened at that point. Yeah.